Hello, and welcome back to Science with Sully. Today, I'll be discussing GQ, G protein coupled receptors. There's a few things that we should discuss about GQ coupled receptors. They're abundant in smooth muscle, and smooth muscle is found in that lining our veins and arteries. So, it's safe to say if it's lining our veins and arteries that it's going to control blood pressure, and it will. It will control the size or the resistance of the veins and arteries, which in turn controls the pressure. So if we stimulate these receptors in our smooth muscle, they are going to raise our blood pressure, which means if we inhibit them, that inhibition is going to lower our blood pressure. So let's see how this works. First, we have our cell membrane. And we have our G protein that is going to make a couple of passes through our cell membrane. 1, 2, 3 passes. And it's going to have its alpha and beta gamma subunits. Alpha, our beta, and our gamma. And our alpha subunit is Q coupled. In this inactive form, our alpha subunit has GDP bound to it. So what's going to happen? Well, like with other receptors, we have to have a start point. Our start point is, like with the other ones, our ligand. Our ligand is what's going to kick everything off. So when our ligand activates our G protein, as we've seen before, our alpha subunit and our beta gamma subunits dissociate. And our alpha Q, and as we know, it is now active, so it has a GTP. So where will the GQ unit go? Well, there's a protein in our cell membrane called phospholipase C. Phospholipase C. And this is an enzyme that cleaves, or lipase means to cleave, phospholipids. Well, there's a phospholipid called PIP2. And we'll draw one right there as a squiggly. So PIP2 goes to phospholipase C. Well, we need to keep in mind our alpha Q subunit is stimulatory to phospholipase C. Phospholipase C takes our PIP2 and converts that, or breaks it, into diacylglycerol and IP3. So when our G protein is stimulated by our ligand, we are increasing the concentration of DAG and IP3. DAG is used as an intermediary in other reactions, but for this reaction and video, we're going to focus on IP3. What's IP3 going to do? Well, down here in the other part of the cell, we have our smooth ER. And in the smooth ER, we have what's called an IP3 receptor. So you could guess that IP3 is going to go to this receptor. Let's move that arrow over a little bit. and it's going to activate that receptor. When the receptor is activated, you may already know that calcium is very high in the smooth ER. So let's draw calcium a different color. Calcium will be green. So we have calcium, and we've got high calcium in the smooth ER. Well, the IP3 receptor is actually a calcium channel. It's a hole, and it's closed unless IP3 binds to it. And when IP3 binds to this channel, it shoots open, and the concentration gradient is higher in the smooth ER and lower in the cytosol, so calcium, without needing any energy, is able to move into the cytosol and outside of our smooth ER. Well, what does calcium do? We should know that calcium, and we'll note this up top, note calcium, high calcium in muscle results in contraction. So the calcium is going to start to increase in the cytosol. And when the calcium increases in the cytosol, we have contraction. When contraction happens, our blood pressure increases. Well, we need a way to move this calcium out of the cytosol. You don't want it in the cytosol forever. You want to uncontract eventually. So we have another receptor. And this receptor down here in the smooth ER is a calcium ATPase. So ATP is converted to ADP, and when that happens, it is able to bring our calcium back in and bring our calcium levels back up in the smooth ER while lowering calcium levels in the cytosol. And the calcium that begins to rise is this calcium over here for when the reaction happens again. Receptors such as these in smooth muscle are generally thought of as our alpha-1 receptors. And when stimulated, they raise our blood pressure. Also, another important part of calcium. Calcium causes our muscle contraction, but calcium does another thing. As the cytosolic calcium continues to rise, and we'll draw another protein right here, so the calcium comes through, and so we have our increasing calcium out here. Well, that calcium can stimulate another protein up here, and that protein is protein kinase C. Protein kinases that are protein kinases phosphorylate proteins. So calcium goes to protein kinase C and is stimulatory of that. And in turn, protein kinase C is now stimulated, and it is going to increase protein phosphorylation, which will activate and inactivate some proteins. And so it's easy to think that protein phosphorylation is the result of both GQ, which is this, and GS receptors are also involved with increasing protein phosphorylation, while our GI receptors decrease protein phosphorylation or kind of acted as the brakes in this reaction. I know this is a lot, but if you watch the video through and through and follow along on your own, you can really see that it builds and builds and builds as we go through and go down. I hope this video is helpful for you, and please remember to like, subscribe, and follow along for more Science with Sully videos.